Hello everyone, in this lecture we are going to discuss about nucleus. So this video is a continuation for the structure of eukaryotic cell part 1. So nucleus was first discovered by the scientist Robert Brown as early as 1831, and the scientist Fleming given the name chromatin after he stained the nucleus with several dyes. So here, if you want to define the nucleus simply, so for the entire organism body, the organism body do have a lot of systems, right? So in order to control those all systems, in order to control the metabolism at organ system level, the brain actually controls all kind of systems in the body, right? So similar to that, in cellular level also, the cell do consists of some signaling reactions, some signaling pathways to occur in the inside the cell, and some other metabolic reactions actually occurs in the inside the cell. For example, DNA replication, transcription and translation are uh, some kind of mechanisms which must occur inside the cell in order to make the cell, enable the cell to be alive. So that kind of DNA replication and transcription, translation, which are very very important for the organism cell, so those kind of mechanisms are actually controlled by the brain called nucleus. So the nucleus is actually known as a cell brain, as it acts as a controlling machinery for the for controlling mechanisms like replication, transcription and translation, and the signaling pathways in the cell. So the interphase nucleus, which is the nucleus of a cell when it is not dividing, is known as interphase nucleus, as highly extended and elaborate nucleoprotein fibers called chromatin, nucleomatrix, and one or more spherical bodies called nucleoli are present. So the space between the two layers of the nuclear membrane, so the nucleus is bounded by a membrane known as a nuclear membrane, and this nuclear membrane consists of two layers. One is outer membrane, another one is inner membrane. So the space between two membrane is known as a perinuclear space. So here the perinuclear space forms a barrier between the material present inside the nucleus and that of the cytoplasm. So as I have mentioned, that nuclear membrane consists of two membranes. One is outer membrane and inner membrane. So in case of outer membrane, it is usually continuous with the ER and also bears ribosomes on it. And the nuclear membrane, when you observe the structure of a nucleus in detail, the pores called nuclear pores are actually formed by the fusion of two membranes. These are the passages for the movement of the RNA and protein between nucleus and cytoplasm. So when you see the structure of nucleus in detail, so here you can see actually, so this is the in detail structure of nucleus. So this is the nuclear membrane. This whole portion which you are seeing is the nuclear membrane, and the nucleus consists of two membrane. One is outer membrane here, which is present here, another one is inner membrane. Okay, so this outer membrane which is present here, it is actually continuous with the endoplasmic reticulum, and also it also consists of nuclear envelope for protection, and these spaces which are present here are known as nuclear pores. So these nuclear pores act as passage for the transportation of proteins between the environments of inner nuclear environment and between outer nuclear environment. So this nuclear pore and perinuclear space serve the purpose of transportation of proteins between the two environments of nucleus. One is intracellular environment, which is inside the nucleus, and other one is extracellular environment of the nucleus. So here you can observe the condensed form of chromosome known as chromatin are present inside the nucleus. So this chromatin are of two types. One is heterochromatin and another one is euchromatin. Hetero and euchromatin are present here. So this is the in detail structure, and the nucleus, inside the nucleus you can observe another organelle known as nucleolus. This is the organelle for the nucleus. So this nucleolus have the application for the synthesis of ribosomes and RNA molecules which serve the purpose of translation. So as I have mentioned, the endoplasmic reticulum here. So the outer membrane is continuous with the endoplasmic reticulum, and also this nucleolus enables the synthesis of, or it actually serves the purpose of synthesis of ribosomes and proteins. So this is the in detail structure of the nucleus. Yes. Yeah. So these nucleopores are formed by the fusion of two membranes. These are the passage for the movement of RNA and proteins molecule between the nucleus and cytoplasm. So here the nuclear matrix or nucleoplasm contains nucleolus and chromatin. So the nucleoli are the spherical shaped structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm, and it is not membrane bound structure. It is a site for the active ribosomal RNA synthesis. So as we all know that RNA and ribosomes actively participate in the process of translation, as the RNA and as the ribosomes get attached with the RNA during translation and forming a polyribosome or polysome. So this complex serve the purpose of protein synthesis. So as this kind of ribosomes and RNA molecules are actually synthesized from the nucleolus, so that is the reason the outer membrane of the nucleus is actually continuous with the ER, which is a rough endoplasmic reticulum. So here the most important point here, the cells which consists of a rich amount of nucleolus are those cells will actively participate in the process of protein synthesis. An interphase nucleus has a loose and indistinct network of nucleoprotein fiber called chromatin, and this chromatin is a condensed form of chromosome, and this chromatin do consists of some basic proteins called histones and some non-histone proteins as well as some RNA. A single human cell has approximately 2-meter long thread of DNA distributed amongst its 46 chromosomes. So every chromosome essentially has a primary constriction which is known as a centromere, on the sides of which a disc shaped structure called kinetochores are present. So these kinetochores are very essential for the attachment of spindle fibers during cell division. So based upon the position of centromere, the chromosomes actually divide into the following types. One is metacentric, submetacentric, acrocentric and telocentric chromosome. So if you see the structure of a chromosome, so these are the chromatids, and this will be the centromere or secondary or primary construction, and at the end of the chromatids you can see the telomeres. So based upon the position of this centromere, the chromosomes are divided into the following types. In metacentric chromosome, the centromere is exactly present in the middle, dividing the two chromatids into dividing two chromatids in equal length. So that kind of chromosomes are known as metacentric chromosomes. So in case of submetacentric chromosomes, the centromere is little away from the chromatids, uh, which leads to the unequal length of the chromatids. This is known as submetacentric chromosomes. So in case of acrocentric chromosome, the centromere is somewhat far from the chromatid. So this structure look like presence of only single chromatid in the structure. Those known as acrocentric and telocentric and acrocentric somewhat similar. Sometimes a few chromosomes have a non-staining secondary structures constrictions at a constant location, and this gives the appearance of a small fragment called the satellite. So these are some important a simple outline of nucleus of an eukaryotic cell. Thank you.